Well, good morning once again and welcome back. We are at the, Carolyn, okay, excellent, welcome Carolyn. Yes, the microphone is here on this computer, the microphone is on the, incorporated to the,
When you hear the recording, the sound is not clear. You stop from.
you're not hearing well, the sound?
It was better this last one than the previous one. Yeah.
Better, but still low, huh? Okay, so this is actually the microphone here on the side of the camera. It's not as high quality as the other one. I think the other one is more professional. So maybe I'll try to speak a little louder and get closer. Hopefully this will work. Sometimes just a few inches is sufficient. Carolyn, we have some handouts here, sorry. Okay.
Meeting a Rotary, Public Square Rotary today, actually, about
Okay.
11 o'clock.
All right. Okay, we begin as always with a little prayer in the name of the Father, the Son, and the Holy Spirit.
Amen.
Blessed Trinity, we love you, we adore you, and we thank you for all your many blessings. We ask that you be with us today, as always, as we seek to understand nature at a deeper level, including our own human nature. We pray for all your people, especially those who are in most need of your divine mercy at this time. We ask all this in the name of Christ our Lord.
Amen.
Amen. Father, Son, and Holy Spirit. All right. Questions from last sessions or comments? Anything at all? No? All right. Let's see. Oh, there's another, Carolyn, sorry. This is the outline for today's class. Thank you. Today's lecture. And, okay, I think most of you responded already to the email about, the email about recording. Carolyn, I need you to look at your STU email, okay, and respond to that. And also changing the lecture in November from the 17th to the 3rd. And most of you are available. I think one student is not, but the majority is, so we'll go with that because they changed the prison ministry on us, and unfortunately we don't have control of that, and I'm the only one that is committed to that for now. Plus, the 10th of November stays as it is with the lecture. Okay, so bottom line, we'll have lecture November 3rd and November 10th, and not on the 17th. And I think there's also at the end of October, there's lectures, so there are continuous lectures coming up every Saturday. Then there'll be a break on the 17th.
Next Saturday
Okay.
we don't have.
No. Next Saturday we don't have, correct? Yes. All right. Anything else from housekeeping? No.
I have
Yes.
a question for the second course for the book. I know you talked
Yes.
about the summary.
Thank you.
When ultimately should we be done with that? What is our plan?
Okay, so that would be first or second week of December. I don't have it exactly, but it's in December. All right. And I send you the link to the book on Being a Scientist. Did anyone not get it? No, it should be in your STU. It must be in your STU emails. Yes. All right. And so you have the link there. Has anyone tried going in yet and downloading it? It worked? Okay. Yeah. So the link works. And then you understand the instructions on just a brief summary of the chapters. The chapters, so sometimes it's like 2 or 3 pages. And so they're very brief chapters, really. So just 1 or 2 paragraphs is sufficient, and you can do them sequentially. You don't have to do a single page for each chapter. You can just say chapter 1 and give it the paragraphs, and then continue chapter 2 and 3 and so forth. It's up to you, you know, how you format it, but you don't have to devote a whole page to each chapter if it fits in half a page.
The video that you sent to watch, the post video, it's same. When I open it, I can't see the video,
Cannot.
the post one.
The same as here, right?
Yeah.
Yes. I think because so that's the TED Talk. Yes.
Yeah, that was hard.
Did anyone get to see it? It's strange. I don't know. I think the format is not a YouTube format. It's something else that I think is called Vimeo or something like that. So it's a different format. In some laptops it opens, some computers it opens, and in some it doesn't. Frankly, I haven't had the chance to go to IT here, our tech support, to see what's going on. But just stay tuned, and I'll try to see if we can solve that technical issue. All right, so that everyone can see the talk. Worst case scenario, I think I can send you the name of the person or something like that, and then you Google it and see if you can find the video through your own computer system, through the OS, the operating system. Okay, so, well, very briefly then, we were looking at, first we saw the evidence for evolution, and then last time, the origins, focusing on the origin of life, organic life, and the possibility of the beginning of life from inorganic material, mostly the gases and the substances that were around in the primitive atmosphere, through a series of processes, including cycling of like the shore coming in and out, and hydration and dehydration. Those cycles would serve as kind of an engine for chemicals to react to one another and to polymerize, etc. Basically, it's an increase in organization and complexity that can go on. But keep in mind that this is over millions of years, all right? Millions of years. So imagine the shoreline, the waves coming back and forth on the shore over millions of years in millions of microscopic locations throughout the universe, throughout the world. So when you start adding those stats, okay, the probability that this could have happened is a real probability. Yes.
I put in, I hope you got my homework because I didn't get your response. But,
yes.
Those last, that sheet where you had
yes.
the
7 steps for organic life. Mm-hmm.
I don't know how other people were about that, but I had serious trouble about understanding from 4, 5, 6, 7, 8. I mean, it just
yes. Yes. I can see the challenge because it was very technical and very chemical, if you will, all right? But these are processes that are well known in the lab. So I really wasn't expecting all of you to understand in depth what was going on. I was basically presenting the evidence for the possibility of the development of organic life from inorganic material.
okay. Well, I got the first 3, but the last
yes.
ones
yes.
I had real
yes, because the language gets more and more technical. Yes, it does. Where there will be another chance at looking at this a little bit is next semester when we talk about the beginning of human life, all right, the beginning of life issues, because there we'll go over at least ontogenetically the development of the early embryo from fertilization forward. And it will give you a chance to understand a little bit more the chemical and the biological background for this. Okay. All right. Anyway, we can go over that at the beginning of the spring semester, just to like a review. I want to move forward now with the issue of variation, because variation is where selection acts, right? So natural selection acts on variation, on variance. In fact, you may recall I mentioned that Darwin did not really make a distinction between variation and species. He doesn't actually accept species. Of course, the concept was fluid, was not fully understood down to the biochemical level because simply those tools were not available yet. And remember, it is Meyer who comes up with a biological definition of species, right? which was several decades later, because Darwin is in the 1860s, 1870s, and Meyer is already in the 1900s, 1920s, 1930s. Okay, so there's about half a century at least of difference of time. But Darwin is very big on variation, on variance, and he emphasizes, he makes a big case for variance because he understands that evolution acts or selection acts on the variance. So some variants we can say that are more fit than other variants. But variants, technically speaking and biologically speaking, still belong to a single species. And that was the great contribution, in my mind, of Meyer, that we can still accept species and retaining the reality of variation, of biological variation. So that's kind of an introduction. Now we're going to go a little more in depth into it, and hopefully after the break, we'll look at the forces of evolution, which are mutation, migration, drift, and then the actual natural selection. Natural selection, we'll devote a lecture to that. So we'll cover 3 of the 4 forces of evolution that drive evolution. Okay, so if you don't have any questions, let's go forward then. We're going to look a little bit at quantitatively, so together, quantitative and qualitative, all right, how they interact, because populations are both quantitative and qualitative. Populations are of particular species, which means that they are qualitative. We're talking about a population of rabbits, not a population of foxes. So there's a qualitative difference between a rabbit and a fox, right? One is a herbivore, the other one is a carnivore, for example. So they have different lifestyles, if you will, different habits of feeding. But also quantitative. Quantitative means somehow the numbers involved, the numbers. So we need to look at populations and then population range. What do we mean by a range? Statistically and also realistically, what is a range? And that's where we're going to see the variation in the range. Then what we mean by polymorphisms, so we need to get into genetics a little bit. Polymorphisms mean different shapes, variety of shapes, basically. When we get a little bit into genetics, we're going to look at what is known as quantitative traits, in contrast to Mendelian genetics, which is all or none. There are no range in Mendelian genetics. And finally, Darwinian evolution with the forces that drive the evolutionary process. So let's go forward. Oops. Why is it so loud? So when we talk about variation, and for now, keep it within each species, within a species, all right? Variation occurs at these 4 levels, at least at these 4 levels. At the morphological level, the physiological level, molecular and behavioral level. And I'll give you examples of each as we go along. But there's variation at least at these 4 levels of living organisms. And so selection can act on any one of these levels, or typically it's a combination of them. Variation is so prevalent in nature. Sometimes is very graphic, is very visible, all right? And other times it is, for example, the difference between a male peacock and a female peacock, which is called a peahen, right? We can see something that is very obviously different in morphology, in shape. Peacock. Got it backwards. CK and peahen. All right. just in case people have not seen a peahen. Okay, this is a peacock and this is a peahen. So morphologically, for example, you can tell the peacock is much more exuberant in colors. Behaviorally, the peahen does not flare her tail. That's a behavioral thing. In addition to the ornament, the exuberance of the tail itself, okay? The color is more drab and so forth. This is due to a type of selection that is not natural. It's called sexual selection, because natural selection implies randomness, but sexual selection is not random, it's oriented. The peahen is selecting on the male, right? On the peacock, and that's why the male becomes more and more exuberant that way. Okay, so that's an example of a very obvious morphological variant between the male and the female peacock. Happens with many birds. In some cases, it's just the opposite. You cannot really tell them morphologically. Let me finish just a thought for a moment. Other birds, the male and the female, are morphologically identical, practically identical, to the point that you actually have to send a feather away for DNA testing to determine which is the male and which is the female. Okay, that happens in birds and that happens in fish also. Interestingly, how you have both extremes. Some species are obviously varied, and other species are obviously non-varied at the naked eye level. Okay, doctor.
I don't know if the screen will be better because it's.
Oh, I'm sorry.
projected on
Thank you. Thank you very much. I can take care of that technicality. Thank you very much. Okay. Okay. It's a smaller screen than the other place, but at least it's a screen. All right, so that's just an example of variation between male and female within the same species. At the physiological level is the chemistry, if you will, all right? Molecular level, we'll get there, and behavioral, obviously, the way they go around nature, feeding and mating and sleeping and running away from aggressors and so forth. Variation is so prevalent that it occurs even in identical twins. So I want to use the extreme case of identical twins to kind of prove to you variation, all right, subtle variation at the level of identical twins, and therefore, if it occurs in identical twins, then obviously it occurs in every other living organism, right? Okay, so I'm using the extreme case. To explain twinning, we have to make a difference between fraternal twins and identical twins, right? So fraternal twins are also known as dizygotic. In other words, they come from two zygotes. And how do we get two zygotes? Well, it just means that that woman ovulated matured two eggs in that ovulation, simultaneously. And typically they're on opposite ovaries, right? So they get fertilized. And because there's millions of sperm around, then whatever egg is there is likely going to get fertilized, right? So fraternal twins come from two eggs that were fertilized by two sperm, therefore two zygotes form and two embryos developed, okay? And they can actually have separate placenta or they can share a placenta depending on when the split happened. No, sorry. In dizygotic, they always have separate placentas because they are two individuals from the start, right? And therefore, because it is the sperm that determines the sex, in fraternal twins, one twin can be a boy, the other twin can be a girl, or both can be boys or both girls, but it's a standard average because two sperms are fertilizing two eggs, right? And the only thing they have in common is that they were gestated together, basically. and that they have same parents, common parents. Identical twins, on the other hand, which are also called monozygotic, the word is significant. Mono meaning single or one, zygotic, zygote, a reference to the fertilized egg, the first cell of the embryo, right? There is one egg and one sperm. So it starts as one individual, egg and sperm at the zygote level, which at some point within 14 days, for the human, 14 days of embryonic development, the first 14 days, the first couple of weeks, that one embryo split into two because there is mitosis going on, there's cellular division. And if that little embryo loses what we would call cellular integrity, then there could be a radical split. It could be at the two-stage level, the two-cell stage, or the four-cell or eight-cell, because they're duplicating at every point, right? They're doubling their number. Even the blastocyst and even the gastrula, the early gastrula, because implantation happens between 5 and 7 days, and then it goes into the gastrula stage, that embryo can split in half, and two individuals will continue growing, gestating to term, and identical twins are born, right? And they're identical because they come from a single egg and a single sperm. So in principle, they have the same DNA. So they are supposed to look alike, very much alike. And they have essentially, I say essentially the same DNA because there could have been some mutations that occur along the way, along the gestational stage, that will make slight differences in their morphology or physiology. Okay. But even identical twins are not exactly identical. They're not really 100% identical. And I'm just going to prove it to you now with a set of twins here. Claire and Laura Newing. All right. So Claire and Laura Newing, I think they're both teachers, and they're identical twins. But they're not really 100%. And it takes kind of an expert eye to find the differences between them at those four areas, at the morphological, physiological, molecular, and behavioral level. All right, but this one picture will show us that if we are observant enough. For example, at the morphological level, okay, meaning their shape, they really look very much alike. But look at this line here. I cut the photo there on purpose. All right. It turns out that Claire is slightly taller than Laura. Just very slightly. Morphologically, what is the longest bone in the body? The femur is typically what determines our height. Okay, the femur is the longest bone. And so kind of from the trunk up, we kind of have more or less the same size, but the legs is what varies most in the human by way of tallness, which is a range. And so it seems like Claire's femur may be slightly longer than Laura. Why? Because it's nature and nurture, right? It could be that when they were both embryos, Claire just happened to get a little more calcium, who knows, whatever, stretched that femur a little bit more, just a few millimeters at the embryonic stage, which then continued along that pathway and just made Claire slightly taller. So that's just an example at the morphological level. You can think of other examples where there is that interaction between nature and nurture, because even if they were actually fed the same food, they didn't really eat each other's food, so that food would also have an influence on the growth, especially in the early years, how much milk they receive, down to the milliliter, if you will, because that all impacts the biochemistry of the body as the body is developing. So we can see that when we add the dimension of nurture, right, that could make slight variation within the morphology of these two ladies. How about the physiology? In other words, the functioning of their body, their chemistry, which is tied in with the molecular, but see, the molecular is more internal, cannot really be observed without a chemical analysis, biochemical analysis. We can do blood typing, for example, and whatnot. But physiology is some manifestation, is what we call the phenotype, or some characteristic that is expressing, right? Some characteristic that is expressing. Well, they definitely have freckles, correct? They have freckles. So this is Claire on the left, and this is Laura on the right. Which one has more freckles? Let me just maybe magnify this a little bit. Right? Which one has more freckles? It's pretty evident that Laura has more freckles if we look at the nose, if we look at the cheeks, but mostly on the nose. So that's a physiological difference between the two of them, because that is melanin, and this gets into the molecular. Melanin is the pigment of the skin. And so whatever mechanism, whatever pathways are producing that melanin, it seems that those pathways are a little stronger in Laura than they are in Claire, because she's exhibiting more freckles than her twin sister. So that's you understand the physiological level is some characteristic that is expressing, that is letting us know a biochemical pathway that is occurring even during their embryonic stage and the early childhood and so forth, as they develop the pattern of freckles. Okay. Molecular, again, molecular is understood that if there is a variation in the physiology, then there also has to be a variation at the molecular level behind it, behind the scenes, if you will, underneath the skin, literally, to produce that phenotype, right? So understand that the molecular pathways for the production of melanin somehow are a little stronger in Laura than they are in Claire. So that is truly at the molecular level, literally behind the scenes, below the skin, in her metabolism, in their metabolism. And this could happen early on in childhood and so forth. Okay, how about, I mean, also, again, nature and nurture. It could have been that one of the two, because melanin is also stimulated by light, by sunlight. It is logical to assume that they did not both have the exact same exposure to sunlight for every day of the years of their life. Let's say they're in their 20s or whatever, you know, for those 20 years. There could have been a slight variation. Maybe one day one of them had a stomachache and didn't go out on the sun that day or whatever. See what I'm saying? So there will be individual variation according to the microenvironment of the development of these two individuals over the years. Finally, at the behavioral level. This one is actually so obvious that many people miss it. Exactly, that's behavioral. Yes, that's exactly right. Claire, for whatever reason, chose white that day, and Laura chose blue. So that's a behavioral difference that is right there, okay? Some have said that maybe Claire is right-handed and Laura is left-handed because they're holding the page on opposite hands, right? But that could have been a camera setup for whoever was taking the picture. They said, put your diploma on opposite sides so that it shows better on the photograph. So that could be staged. We don't know for sure if they're right-handed or left-handed. And I don't even know if identical twins can be independently right-handed or left-handed. Do you, Dr. Benchance? I don't know. That's an interesting question. I wonder because it has to do with the way the brain works, the writing center, where the writing center is. Okay, and you know there is that the cross section, not cross sectioning. That's the word I'm looking for. Or cross pathway, for example. What happens on the left of our body is typically controlled by the right of the brain, the right hemisphere. So I'm left-handed, which means that the writing center for my brain is on the right side, okay? And people who are right-handed, which is the majority of the population, about 94% in the United States, you'll have your writing center on the left lobe. It's just where it's distributed at the beginning of the centers when they develop. So I don't know, maybe someone wants to look that up as just a curiosity. If identical twins can be opposite-handed writing, the center could be on opposite sides. And that would point to some kind of nurturing happening that there's fluidity on that writing center. And there's some kind of external mechanism that triggers where the center goes, either to the right or the left hemisphere, no? I'm just kind of thinking out loud here, what would trigger that center to develop either on the right or on the left-hand side?
And
Yes.
some parents say, I don't want you left-handed. And this one should be
Sure.
right-handed, and that would shift that.
Yes, and that was very prevalent, but there is still a natural tendency for left-handedness. And I'm glad you bring that up actually, because even within right-handedness and left-handedness, there is also a range. There's a range so that drastically the mid-range for that is that some people are totally ambidextrous, okay? And even more radical than that, they write in opposite directions. It is said that Da Vinci had that quality, that he could write from the right hand, like most of you are right-handed, from, so normally a right-handed person writes from left to right, okay? But we left-handed, we still have to write from left to right because it's a right-handed society. Now, way back last century, at the time of the ink blot and the fountain pen, which I was growing up with a fountain pen, and I remember my desk as a kid in school had a hole for the well, for the ink well and the ink bottle. So we dip the fountain pen there and write. And I always had this finger here black from the ink because I would smear on top of what I wrote as a kid. And so the nun would come around and crack my knuckles and so I have a horrible handwriting because of that. Both my parents were left-handed, and they tell us, my brother and I, that but my brother is right-handed. At any rate, my parents told us that both their parents forced them to be right-handed, okay? But I inherited the left-handed, so I became left-handed, or I was born left-handed, and my brother was born right-handed. So it's hard to tell. I really don't know the genetics of it in detail, but I know that there is a range, and some people are extremely right-handed, and the opposite is some people are extremely left-handed, so that you really can mess them up if you try to change them. Typically, left-handedness was, any variant that was off the norm was seen as an anomaly, but it had moral implications, to the point that even the word in Italian, sinistra is left-handedness, and in Spanish we say siniestro, right? A left-handed person that's where we get the word sinister, is a sinister person. So left-handed person is a sinister person. That's the kind of siniestra, it's in Italian, okay? So it was seen as the work of the devil, that this kid was possessed by the devil somehow. And that's why the parents insisted on taking that left-handedness away, all right? But we know now, psychologically, that if the kid is very left-handed, that it can traumatize the kid and really affect him or her. And so it's better to leave them alone, perhaps mildly encourage them to become right-handed, simply for convenience, for practicality, because it's still a right-handed society. It's the majority of the people. So for example, when there used to be wires to the phone, all right, the wire was always in the way, the cable, the phone cable was in the way for left-handed because it's on the opposite side. Doors for us open on the opposite side. Everything for us left-handed, it's all backwards. Yeah. And even to the point that statistically, in the population of left-handed, we have a shorter life expectancy because we run into things. I can tell you how many times I've run into a door, and one time at Purdue, I actually slashed my forehead with a door because we run into things, because it's a right-handed society naturally. Mm-hmm. For example, I would love to drive on the left hand of on the right hand of the car, you know. The opposite side of the street, for example, like Britain, British driving on the opposite side of the street. But so anyway, these are things that are just part of society, but the left-handed and the right-handed here could have been staged, we don't know. But it could be interesting.
In evolving, do you find yourself training yourself on right-handed?
Yes,
as it goes on?
At this point, I think the toughest part is the early years when one is adapting to so many things, right? And so that was the toughest part, really. At this point, even with computer, my handwriting remains lousy, but now I touch type, so it's much easier for me to touch type things than to write it out. In fact, you'll see my scribbles are hard to interpret, and sometimes I have to go over the letters because I do it incorrectly. So when I give you back the, which I have it here, the homework.
You use the mouse on the right hand.
Sorry? Yes, I do. So I just become accustomed to use this because this was, of course, the mouse came around much later than my just adapting to a right-handed society. So, and in fact, on purpose, I try to use my right hand as much as I can. On purpose, but the writing, that is really, what do you call that? That's where I draw the line. Because if my left-handed is right, if my left-handed writing is atrocious, my right-handed writing is totally illegible. Okay, but it's also a question of practice. I still remember doing the circles and doing the wiggles and all that when we were kids, trying to keep it inside the line. Yes. Okay. All right. So anyway, you understand that these four levels, the morphological, the physiological, the molecular, and the behavioral, variation can happen. Variation can happen at these four levels. All right. Okay. And if it happens in identical twins, in a subtle way, it happens in all individuals in more or less subtle ways, depending on what we're looking at, what characteristic we're looking at. All right, which, by the way, those characteristics are technically called traits in genetics. These are genetic traits. That's actually getting a little too long.
I have to just say,
Yeah.
it's funny as I'm listening to this, I have twin nieces
Oh, yes,
and they're identical twins.
yes.
One has a freckle,
Uh-huh.
so I always have to look at it when I first see them.
That's the
Or
freckle.
I observe and I was doing this before I realized that I would observe their behavior
Mm-hmm.
because one is a little more quiet than the other, and you just see the subtle differences and
Yes.
you know who's who.
Mm-hmm. Yeah, you see, so, and that's mostly behavioral, right? And even when you think about that behavior early on, even the subconscious behavior of lactating and whatnot, that could have caused later some other morphological differences simply on their individuality. Right? So Darwin was emphasizing a lot that individuality. He was really focusing on the variation of the individuals. Emma.
When I did ultrasound,
Yes?
I noticed that when I did twins, they had a membrane. What is that membrane for?
So where was this membrane? You're talking about?
Separating the
Yeah.
twins from one another. It was like
Okay.
kind of like
So that could be the chorion or, okay. And these were identical twins or
no,
dizygotic?
not
No?
identical twins at the same
Okay.
line.
So that could be the amniotic sac, no? It's probably the
They're
two amniotic
separate.
sacs.
Exactly. They're separate, they're two separate.
Bags, yeah, right. So, okay, so those were dizygotic, and because in monozygotic twins, depending when the split happens, they could have separate placentas or they could actually share placenta, all right? Depending on when the twinning happened. But those cases are probably most likely dizygotic twins. When you think of it, fraternal twins, non-identical, they each have their own embryonic sac, right? The amniotic sac, and it's just the two sacs going against each other like that, full of fluid. So it looks like a single membrane, but it's actually two, right? Make sense? Mm-hmm. All right, so let's look at variation. In nature and in all populations, and what we call, because variation then gives us a range, right? Again, to recall the definition of population, remember population is two or more, what? Individuals within the same species, right? So you get the concept that it's the same species, all right? So it's the same animal, plant, fungus, whatever, is the same species. So here we have different populations of different species, right? Definition. So here's a population of how many tigers? Lions. Is this a population? That's a population of 4 lions. Don't miss the one in the back here. That is camouflaged. It's probably the mother. She doesn't have a mange, right? So the mother and the cubs. I think it's actually called a pride for lions and felines in general. All right, so we have a small population of 4 lions, all the same species, different ages. So there's an obvious range in age, for example. There may or may not be a range in sex. This is definitely a female because she is mature already and she doesn't have a mange like the male, all right? And so most likely she's a female unless it was a mutated male that didn't get a mange, but that male would be very unlikely to mate because it's at a definite disadvantage on the show. At any rate, there are 3 cubs here, and we can't really tell from the photo if they're male or female yet, but at least the adult is a female. But then variation within the cubs, and we can assume it's reasonable to assume that they come from the same litter, right? So they're about the same age, so there's no variation, let's say, in the age of the cubs. But how about, they look pretty much alike in their pattern, but I found a slight variation in a pattern that may have a significant difference when it comes to natural selection, or let's say sexual selection. This one actually, sexual selection may be a little more clear. I'm focusing on these 2 cubs. This is a blow up on the 2 cubs. If I blow it up anymore, it starts losing, it becomes a little fuzzy. But I just wanted to concentrate on the ears for a moment of these 2 cubs. You see, this one seems to have a darker spot on the ear, and this one doesn't have a darker spot. See up here? This one has a dark spot there. This one is missing that dark spot. Okay, now that may be significant because this spotting is not just by chance. Typically, traits or characteristics in nature have some kind of function. For example, whereas the cubs seem to be unaware of the photographer, the mother seems to be aware of the photographer, right? Or at least of the camera, because she's looking right at the camera. I look at least this ear is what compared to this ear? This ear is propped up.
Right. Like
Is propped forward, exactly. So she's got this ear focused on the photographer. she's got this ear focused on the environment on the side. And it's very telling when we look at the detail because she's looking out for her cubs. I mean, we can imply from this picture a lot. It's reasonable to assume she's focusing on this other individual who is definitely not a lion or a lioness, okay? And it's not part of her pride. And she has a big snout, so she's probably smelling something that is foreign. Hopefully the photographer is downwind, but even so, some smell will get to her because they have a potent snout. But definitely this ear is propped forward, which they can do that. We cannot, we've lost that characteristic because it's not that significant for us. But many animals do, mammals flip around their ears. You see the horse, right? Flipping their ear forward or sideways. This one is not forward. So when those ears are forward, let's say even both of them or just one, may give a different, slightly different signal between this ear and that ear. In other words, that black spot may have some selective advantage or disadvantage to have it large and small. Okay, my first intuition that comes to mind is just concentrate on the dark and light patterns on the face of this animal. You have a dark parting on the nose, then two dark patterns on the two eyes, and possibly two dark patterns also, again, two dark spots on the ears. So that is generating kind of a V-shaped form that may have some meaning on the face. The pattern of the face may mean something either to the male or to another female. And I don't know, but the larger spot on the ear may mean more aggressive or less aggressive, more vigorous or less vigorous. Is may be equivalent to a peacock with two peacocks, one with a larger fantail and the other one slightly smaller fantail. The peahen would tell the difference, could tell the difference, even if it's just a matter of inches. She could tell the difference because she's focusing in on that aspect. All right, so it could be that subtle, or maybe not. All I'm saying is that there's a range of variation here on the spot of the ear between these cubs, and that may or may not have a selective advantage or disadvantage.
Or can also be
Yes.
camouflage, or when they're hiding together.
Exactly.
It can help the ones that are spotted because they're not noticed.
Exactly.
It's easier for them to
That's exactly right.
find food.
Yeah, that's a very good point. In fact, look, this one over here, I mean, there's a lot that we can tell from this picture. That is their terrain, right, where they're at. And this fourth one, this third cup in the back, is not very noticeable at first. When I ask what's the number in the population, most people say at first just 3, because they're so impressed with the lioness and then the 2 cubs. And this one is indeed camouflage, and the ear plays a part in that camouflaging, right? In the whole composite. Yeah. So I didn't know exactly what was going on here until I think I figured out is the tail of this cub that is somehow mingling with the mouth, the lower jaw of the cub in the back. At first I couldn't tell if this cub was yawning, was holding the yawn with its paw or something. Anyway, so you see my point, this is variation. It's subtle, but real, but it could be very real for them. It could make a difference between survival or not survival between these 2 cubs as they grow, all right? We don't know, but it could be. So that's a subtle one. Here is a very obvious variation on these tulips. These are evidently the same species of tulip, but look at the large variation from very red to very yellow, and just about every tone in between, including some variegation on the veins. So you can see for some it's very obvious, the nervature of the, which is not true nerves, this is just the, they're called veins, but they're not, it's the passage of the vessels or the petal which I've mentioned is in a super adapted leaf, right? But you see my point? From the very yellow to the very red, and every variation in between. So this definitely forms a wide range on the color of this tulip species. Wide range in humans, which, by the way, we are all considered a single population biologically. Humanity, all human, the 7 billion that we are across the world, okay, we are considered biologically a single population. Very different from populations in nature, because you can have various populations of the same species because they're living in particular geographic regions. All right, and they have a range, a geographical range, in addition to the variational range, different ranges. But for the human, because we're so mobile that we can basically span the whole world. So in principle, an African man could mate with an Alaskan Aleut Inuit woman, all right, in principle, and produce fertile offspring. So all humans classify as a single species and a single population because of our high mobility. Okay, so now variation going toward the genetic level, but still the expression of the genes is called polymorphism. Okay, polymorphism, meaning poly, several, morphos, shape. Okay, so several shapes within the same species, which, by the way, the dog is, as you know, an offshoot of the wolf. So the scientific name, Canis lupus familiaris, is actually at the level of subspecies. That will get the dog is a domesticated wolf, essentially, domesticated wolf. So you can see the range in sizes and shapes, right? But all these belong to the same species. So in principle, they can all interbreed and produce fertile offspring. If they can work out the anatomy, basically, then they can all interbreed, all right? And so we get different breeds of dogs. It's another way of saying different varieties. But because they are artificially bred and they were domesticated artificially, then we call it breeds instead of varieties. Variety is more for natural phenomena, a natural variation. Breeds is when we're into artificial selection. For example, the breeding of horses, the breeding of cattle, the breeding of many species, fish, aquarium fish, tropical birds, pet shop animals, plants also. How does that look statistically? Well, the typical polymorphism, and we have many, many polymorphisms, right? Traits are polymorphic. The vast majority of traits are polymorphic, right? Well, most of them, the vast majority, follow what is known as a normal distribution. Now, quantitatively, when we do the numbers and we plot out those numbers, it's going to look like, it's going to give the shape of what is known as a Gaussian curve. Gaussian curve, thanks to Mr. Gauss, who is the one who developed this concept. And it's also known as a bell-shaped curve for obvious reasons. So for example, if we look at the height of humans, well, we have to, first of all, establish a couple of parameters, at least a couple of parameters. We have to establish the parameter of sex. So we're talking about men or women because there is a slight dimorphism between men and women that is natural, okay? Women are naturally a few inches shorter than men, or when men are a few inches taller on average. So that's the first subgrouping that we have to do. The second one is by age, obviously. because as we age, we grow, and then at some point we start diminishing size also, right? Because we start losing calcium, the vertebrae collapse, and we start shrinking. That's the third stage. But once we do that selection, and let's say we're looking at men that are 30 years old here, just to put it out there. All right, so these are all men in their 30s. We're going to have a range of heights of men in their 30s, okay, or even at 30 years, if you will. And normally it's going to look like this. Okay, if they are representative of the whole population, if the sample is representative, it's going to be a normalized curve, where most of those men are going to be, I don't know exactly what is the average height is, well, I think it's 1.8 in meters, but in feet, it's about 5'10", 5'9", something like that. The average height for men, adult men, I don't know, let's say 5'10", approximately. That would be the norm here, the center, okay, the average. And so you would have more or less equal number of variants to either side of the curve until you get to what is known as the tails down here, which are the two extremes, right? So a few men will be very short and one will be the shortest, the very shortest of them all, right? Not being a midget, just normal, because midget is also a condition, all right? There's also variation of range among midgets. So you have the very midgety midget to the very tallest midget. We would expect that to be a more narrow range, right? And then the opposite side of the curve will be the tallest ones. And the tallest tall fellow would be the one who is by far the tallest tall, you know, the basketball player or whatever. And he's way out here. But they are diminishing in number, okay? And that's what gives the bell-shaped curve, or what is known as a Gaussian distribution, statistically speaking. Then you can figure that these bells can be from very narrow, little variation, to very broad, wide variation. Okay, so you can also do a variation on the variations and see, on average, most variations would be kind of middle of the road bell shape. But some bells would be very narrow bells, and other bells would be extremely wide bells, depending on the trait that we're looking at. Okay, let's go forward. Not all distributions are normal distributions or Gaussian. Some are skewed, called skewed. They can either be skewed to the left or skewed to the right. And typically that is pointing to some kind of selective pressure that is selecting out, right? Let's see if it's skewed to the left, which here is indicated as a negative, as a positive skew. What does that tell you? These individuals, the taller ones, are being selected out. There's an advantage, there's a selective advantage to being on the shorter side, being favored by some kind of environmental factor, okay? So that will skew the curve. The taller ones, they stick out too much and they're gonna get eaten or something. The opposite effect can also happen. There could be an advantage to being the taller one, and so that's called a negative skew in the sense that the shorter ones are being selected out. Better to be tall. That's happened even in the human population in Africa. Earlier on, there's one particular tribe, the Maasai tribe, which are very, very tall, all right? And of course, they're very good runners because it's that long femur. Another example of variation, these are called heliconians. These are butterflies that are related to the dragonfly, by the way, but you can see some similarity, but they're not dragonflies. They belong to their own particular species. You can see the range of variation in the pattern also here. There's also what is known as a bimodal distribution. Bimodal means it has two modes, right? Two modes meaning two peaks. And this happens with the height of men and women. If we separate the women from the men, because, so here's the answer. For men, the average height is about 5.75, okay, feet. And for women, the average height is 5.25. So that's quite significant. That's about from 2 to 3 inches. 3, 4, 5, 6. No, to 7, 4 inches. 1, 2, 3, 4, 5. Okay, 5 inches. 5 inches on average. Now you have to think this is a global average, okay? So they also include people in the Polynesian Islands, they include people in California and Iceland, part of the world, where we can think, you know, for example, Northern Europeans, we associate them with being relatively tall and slender and so forth. Okay, but this is just height. This is just height. So this is at the individual level. When we cluster men and women into a single population, for height, we get what is known as a bimodal distribution because of those two peaks. See? So these are variations from the normal distribution. Again, many traits in life are normal, but some are skewed, some are bimodal, and so forth, depending on the actual characteristic and the environmental factors that are influencing it. Okay, so let's continue along the quantitative line here for a little bit and look at some numbers. Very simple, all right, but this is just a very simple review of what should have been learned in elementary school, reviewed in high school. And the only reason I say this is because we get college students that cannot really make this translation between a decimal, a fraction, and a percentage. They just can't do it. Because you know what happens? They pull this out. They pull out the phone. Well, wait a minute, professor, here. Oh, yes, 0.5. Okay, but it is essential as scientists, you'll be getting a master's in science, okay, that you'll be able to make this conversion in your mind very easily back and forth. So even we say 0.5 or just 0.5, because we're always trying to make it simple.
Did we have a sheet on that? Or no?
Not on this one in
No,
particular.
already
This,
has been.
yes.
Okay.
This is because in the bigger picture, I want to talk about this probabilistic system or the probabilistic world or universe. And I've mentioned this before briefly. We are constantly having to go back and forth as scientists between a probabilistic model and a deterministic model. And even Meyer addresses that in the earlier chapters, right? The probabilistic model and the deterministic model, whereby probability, science works on probability, even medicine, the practice of medicine is a probabilistic art, all right? No doctor is going to tell you, well, yes, you're going to be cured with this protocol, or not, you're not going to be cured. No, they'll say there's a 50% chance, there's a 90% chance. So they say most people, now they're just qualifying most people for what it could be a 90, 95%, okay? But a few people means it's definitely less than 50, maybe less than 25%, all right? So science forever is working within a probabilistic model because it's based on accumulated facts, accumulated numbers. But it happens that we're not statistics, right? Each one of us is a determined individual human being. So in front of the doctor, the patient is not a probability. The patient is an actual determined human being. So how to translate that into the real person? It could be that I'm the exception on the statistic, you know, and then I'll skew the average on the norm one way or another. Or it could be that I fall right on the mean of the statistic. But that's the challenge, going back and forth between a probabilistic model, which is how science works simply because it's accumulated evidence, and deterministic individual human or just entities, organisms that are in front of us, whether human or not. And also associated with this, the whole issue of population, when you consider this, for example, is precisely a sample of the population of lions in Africa, or in this particular country of Africa, or this particular savanna. But this is definitely a sample, right? These are not all of the lions that exist in the world today. So is a sample representative or not? What's the magic number for statisticians? Remember when I was in, I took some courses at, when I graduated from biology here, FIU, with my bachelor's in biology. I went to Tampa for a while, wanted to get into USF, and I took a course in statistics there. And the title of the book was The Use and Misuse of Statistics. Because with statistics, you can just about prove or disprove anything you want, depending on how you skew the statistics, right? If you put ideology behind the statistic, you can make just about any argument possible. So the magic number is 100 for the sample. But that's just out of convenience, because 100 is a simple number to work with, right? It's a simple number. But it depends. In the population is, you know, do 100 people represent the whole human race, the 7 billion of us? You'd think that's insignificant compared to the whole human population. And yet, it was even less than 100 people, I think, that were the volunteers for the Human Genome Project, for deciphering the human, decoding the human genome. Including, I think, Greg Venter. I think they also sequenced Watson. Anyway, it was interesting to see who were the volunteers for the human genome, but it's a very small sample of the human population. So there's variation, even when you go into GenBank and you look at that, the 3 billion base pairs that make up the human genome that is sequenced, you know, letter after letter, the 3 billion of them. There are polymorphisms within that sequence that are not shown there. So that has to be accounted, has to be taken into account. Anyway, so the magic number is 100. It depends. It could be that if it's an endangered species, they don't even reach 100. For counting bacteria, that's nothing. So it really depends on the population, the quality of the population, and the species, what that magic number is going to be that is representative, the sample, representative sample. But we're forever dealing with samples, so it's a statistical model. All right, so the mean or the average is this very simple formula, which is a fraction, right? It's the number of individuals divided by the number of individuals. Okay, the sum of the individual numbers, divided by the number of numbers, whatever that trait is. And this is also known as the arithmetic mean or average. There are different types of means. There's also geometric mean and so forth. But this one is the arithmetic. It's a simple one. It's the one that, you know, what's the average height between all of us? That's the arithmetic average. And the range is all the numbers or the observations in a population. That's the range, or the sample. That's the range. So is a sample representative of the range in the population? Variant is each individual, because each individual will have some kind of variation from the mean. Again, staying on the example of height. Well, that's one trait, that's one characteristic, and we all have a specific height, right, at a particular time in our lives. However, that's not the only trait that we have. We also have, let's say, weight. And height and weight can vary somewhat dependently. So they're interdependent, but somewhat independent. On average, a taller person may weigh more than a shorter person unless the shorter person is fatter and the taller person is skinnier. So we can see that those two traits are not totally independent, but they're not totally 100% dependent either. So they're interdependent, right? And so on and so forth with every other trait, just staying with the human body for now, the human organism. So we're all variants. Even if by height, I am exactly the height, the average height for my age right now, all right? I may not be exactly the exact weight for that age, even though if I'm exactly the right height. Or if I'm exactly the right height and the exact weight also, I may not be the exact skin color for that age and that height and so forth. So when you start adding traits, of which we have thousands, and if we go molecular, we have millions of traits, right? Then they're all not going to be exactly on the ideal center of the norm, center of the mean. So we're all variants one way or another. On some trait, in fact, for the majority of traits, we're variants. We can have that intuition, okay? That's what Darwin was focusing on the variants, because he had the great intuition to realize that selection acts on variation. Okay, so we can take it one step further down to the genetic level. And these are three terms that are very important: genotype, phenotype, and genome. All right, these are key terms, and you should at least know what they mean to some level of depth. So the genotype, the genotype is the genetic makeup. It's at the level of the DNA, of course, of each individual, okay? So we all have an individual genotype, which is at the level of the genes and the DNA code, the genetic code. Because there are slight variations in that genetic code, even though we all belong to the same species. Now, the phenotype is the observable characteristics, the traits, is the manifestation of the genotype, which may express or not, because we have latent characteristics that don't necessarily express. For example, we have what is known as oncogenes or proto-oncogenes, which potentially could become cancer genes, but don't express. Thank God. Sometimes they do, then we get the cancer. But if they don't express, we have it in the genotype, but it has not shown a phenotype. It has not shown an expressed and observed characteristic. Now, the characteristic could be at the molecular level, but there has to be some level of expression of the genotype, at least to the protein, to the level of proteins. And so that expression will be observable, again, at the four different aspects: at the morphological, the physiological, the molecular, which is the more sophisticated one, or at the behavioral. It could even be that it's expressed, that phenotype is expressed all the way to the molecular level and the morphology and physiology, but not at the behavioral. It doesn't make a difference behavioral, okay? Those two twins are still behaving the same way on a particular task, on the way they comb their hair or whatever. So you see, it gets complicated when we look at the phenotype. Is the observable expression or trait, but the genotype remains below, underneath, and unless we do really a chromosomal analysis or a sequencing of the DNA, we will not really know the true genotype. But we can estimate from the average, from the GenBank issue, from the sequencing of genotypes of species, and we say they're representative of that species. But we understand that it's going to be variations. Now, the genome, in contrast to the genotype, is the complete DNA sequence, all right, in each cell of an organism. In other words, the entire DNA sequence, that's called the genome, which is shared by the species. So the genome is, if you will, at the global level. So we can talk about the human genome. But within the human genome, there will be variants. So this human genome that I keep referring to, which was a project done in the 90s and was finally completed around 2001, 2002, more or less, to a 99.9999% accuracy, okay? In 3 billion base pairs, there are a few errors there. So it's it's idealized, if you will. That's called the genome, and there's a genome for each species.
I'm sorry
Now,
to be
yeah.
kind of ignorant, but what does the 3 billion apply to?
The base pairs, you'll see that actually in the next slide. You see these are base pairs. You see A and T and C and G. Those are the base pairs. So each one of these 4 letters is called a base. It's just a molecular structure. And they each have a pair. I had mentioned earlier at some point that A always pairs with T and G always pairs with C, right? So we call them base pairs. They stand, the letter stands for a whole word: adenine, thymine, guanine, and cytosine. It's just the molecular structure of these rings. They're molecular rings. This will become a little more clear as we go further into that structure, but not at this level yet. In the spring, when we look at the beginning of human life, we'll get a little more molecular. So the genome, I'm saying, for the human genome is 3 billion base pairs, and it's fairly consistent with a number of other species at the level of base pairs. Just to say that it is very large. The genome itself, it is huge. However, from those 3 billion base pairs, the vast majority don't have a coding region. In other words, they don't really code for any particular protein. All right, it's estimated that only about 3% of the human genome has coding region. In other words, codes for some kind of protein. And the rest, the 97% of that genome, is called just junk DNA. Junk DNA, but in quotations, because it doesn't code for a protein, but we don't really know if there is another function for the DNA that we haven't discovered yet. There's a lot of research being done there. I did part of that. I was involved in that at Purdue when I was doing my dissertation on the structure and function of DNA at the higher order level, the 3-dimensional level. But the point here is that the genome is for the whole species, okay? And each species has a particular genome. Now, some other species could also have 3 billion base pairs, but they will not have the same sequence, obviously. They will not have the same sequence. That makes them a different species. By sequence, I mean the order of these letters. You see this particular order of the letters? And also, we call the DNA sequence to be redundant. Why redundant? Because it's because they are base pairs, and A and T are always paired, and G and C are always paired. If we have one strand of the DNA molecule, we can decipher the other strand. All right, we can decipher the other strand. So there's a level of redundancy there. If we have one strand, if we have deciphered one strand, we can decipher the other strand just by the pairing algorithm. Okay, so this diagram just shows briefly the levels of structure of the DNA, starting from the chromosome level, all right? This is at the level of the genotype. We're focusing in on the genotype. So the chromosomes, which can actually be seen under the microscope with a high-power microscope, the chromosome is made up of coils that are called chromatin. And these coils are actually coils within coils within coils. So they're known as supercoils. When we start unwinding that chromatin, we get into more sophisticated level of coiling in between here, like a spiral. And we unwind further that spiral, we notice that there are these little units called nucleosome. And the nucleosome is like a package that is made up of proteins in the middle, a little protein ball, and the DNA helix, this is the DNA helix, winds around that protein ball 1.7 times, almost 2 times. So think a little bit, let's take a tennis ball and just wind some thread around it twice, a string. You wind a string around that tennis ball twice, okay? More or less, that's kind of the structure. So that's the functional unit of DNA. The functional unit is called the nucleosome. And that's kind of the standard packaging of DNA as we go toward the higher structure, the higher order structure. Or we can start from the opposite end. I think many people are familiar with this diagram of DNA. You see it all over the place, a twisted ladder or the double helix, right? We see this projected. This is already at the 3-dimensional level, because if the helix is twisted, right? If the ladder is twisted, then that is implying a 3-dimensional structure, which starts with the actual sequence and naturally coils this way. So that's the first level of coiling, if you will, is a double helix. Then the next level of coiling is around the protein ball. Those particular proteins are called histones. And they're very important because this is such a standard structure that it occurs in essentially all eukaryotes, all right? And that means that these histones, these proteins are highly conserved. They have kind of the same sequence, very similar sequence of amino acids to make these histones in all of the eukaryotes, which includes animals, plants, fungi. You know, millions of different species have the same basic structure for packaging the DNA inside the cell. So that's why we call the nucleosome the functional unit of DNA, how it's packaged in the cell, in each cell, inside each nucleus of each cell of the body. Okay? So it's very stable, and you can see that it's super wound up, super coiled. It's actually called supercoiling, because these nucleosomes then wind on top of each other, forming this intermediate coil, which again continues to wind, as you can see here, into a higher order structure called the chromatin. And then when the phase is dividing, only when the phase is dividing, do we see there's a highest level of condensation of the DNA material into distinct chromosomes, which for us humans is going to be 46 chromosomes in each cell of the body, or 23 pairs, because they come in pairs. So basically what I'm doing here is I'm taking the genotype apart for you and showing you at the most basic level is a sequence, the base sequence or the base pair sequence, which is where the information is stored in the actual sequence, just like words. I can take 3 letters and recombine them into 3 different meanings. For example, if I take CAT, all right? And in that sequence, CAT means cat, the animal. But if I rearrange the sequence of those letters, if I put ACT, act, means something different. It's not a cat. A cat may act or not act, okay? But it has a different meaning. And if I rearrange it again, I put TAC, tack. It's a different, so the cat may act on the tack. But you see, the same letters rearranged give different meaning. So there is a power in storing information. And this is what we call, this is another one of those elegant moments, how the information is stored in the DNA at the level of this base pairing and the sequence of the base pairs, which translates into the units of proteins that are called amino acids, okay? Let's back out a little bit. So this is just to go to a certain depth of the genotype, and this is the actual genotype, the sequence level here, for each one of us. This genotype may or may not express into a phenotype, which would be then the observable characteristics that we see, the phenotype, what can be seen. All right, so let's look at the phenotype. So the genotype stays in the background, the DNA stays in the nucleus. When the DNA needs to be expressed, it will send a message out to the cytoplasm to produce a protein, and that message is called messenger RNA, which is another molecule very similar, it's complementary to the DNA, but it's a single strand instead of a double strand. And when that RNA is sent into the cytoplasm, then the cytoplasm will produce a particular protein based on that sequence. It will produce either the ACT protein or the CAT protein or the TAC protein, right? Then that protein is already the phenotype because it's an expression, but that's at the molecular level. That may or may not have a morphological expression and or a physiological expression and or a behavioral expression. But at least at the molecular expression, we already have a phenotype with the protein, right? With the protein expression. And that's called the translation from DNA or from the genetic code to the protein. So here's an example of protein expression at the morphological level. The earlobe, right? Whether it's attached or not. This happens to be one of the few Mendelian characteristics, all or none. We either have an attached earlobe or a detached earlobe, the lobe itself, okay? It must be a vestigial thing. There's not known, at least I don't know of any function to it. I don't think women necessarily select on the earlobe. For men select on the earlobe maybe because that's where the earring goes, right? And you notice that of all the species of animals, we are the most selective one because we both select. The man selects and the woman selects, and the vast majority of that selection is going on unconsciously, okay? It just goes on unconsciously. It becomes very graphic, for example, in dancing, all right? There's active selection going on in dancing, all right? But there's the level of selection that is going on subconsciously is amazing. And we hate to admit it, but we are mostly more hardwired than we like to admit with regards to free will. But it just happens, it's part of nature. At any rate, back to the earlobe, there it is. This one is obviously unattached or free. It's not falling off or anything like that, but it doesn't have this little part of flesh there as in the attached lobe. Well, that you can trace it back down to some kind of protein expression of a genetic code, all right? As far as we know, at least as far as I know, this is not produced by different types of food that or exposure to some kind of climatological, environmental issue. So it is truly nature, in this case, is not really nurture in between that dialogue, okay, as far as I know. So babies are either born with a free earlobe or attached earlobe, men and women. I don't know the ratio either, if it's skewed or significantly different. Anybody? More? And I think the free is more prevalent than the attached. Typically, one is more of these all or none characteristics, all right? One is prevalent than the other. Okay, so that's an example of phenotype, how we go from genotype to phenotype, right? The genotypes remains inside the nucleus of the cell, but the phenotype goes out into the individual at some level of expression. At the level of genome, again, we're back to the entire sequence of DNA. So here are the 23 pairs of chromosomes represented. Can anyone tell me, is this a male or a female? This is called a karyotype. Karyotype. Well, the first 22 chromosomes are going to look pretty much alike, except when we get to the 23rd pair. The 23rd pair is either XX or XY, and what makes the male is the Y. So the Y chromosome makes for the male, so this would be a karyotype of a man or a male. If it was XX, right, it would be a female. Because the woman, the female, always produces an X in her eggs, right? But the man will, the sperm, 50% of the sperm will be X sperm and the other 50% will be Y sperm, depending on how meiosis occurred and the separation of the gametes. So depending on which sperm was the one that fertilizing, whether it was an X sperm or a Y sperm, if it was an X sperm, it would make for a female. If it was a Y sperm, it would make for a male. And that's why in the sex, for mammals, it is typically the male that establishes the sex of the offspring. So the first 22 chromosomes are called autosomes. Autosomes. Is that large enough? Can you see it? Yeah. Okay. And the 23rd pair, right, is called the sex chromosome because it determines basically the sex. And there are also what is known as sex-linked characteristics, meaning that especially in the X chromosome, the female chromosome, there are other characteristics that are associated. Those are called, those are sex-linked characteristics, right? That will be in that chromosome, but have nothing to do with the actual sex or the actual reproductive organs, but they're linked to that chromosome. Okay, let's move forward then. Let's look at a little bit more Mendelian. What we have to see here with genetics is the difference between Mendelian genetics and non-Mendelian genetics. Okay, so I'm going to start with Mendelian genetics from Gregor Mendel. This is really what he discovered, these hereditary traits or the inheritance traits or the factors that he mentions. These are typically all or none characteristics. In other words, either they occur or not. They occur or not, all right? And he found these 7 characteristics on the pea plant, okay? The normal green pea. All right, the pea that we're used to, especially for food, is the round green pea. That's the one that we're normally used to. But there's also a yellow pea, and there is also a wrinkle pea. So you see, these are, the peas are either round or wrinkled. They're either green or yellow. The pod is also either green or yellow. So you can have a green pea in a yellow pod, or a yellow pea in a green pod, or vice versa. So they're independent traits or characteristics. Then the shape of the pod is either filled out or constricted, which shows the contour of the pea on the inside, right? So they're full or flat or constricted. That's with regards to the pod. So 2 characteristics of the pod are Mendelian characteristics. 2 characteristics of the pea, of the fruit, are also Mendelian, the color and the shape. Then the height of the plant. This is interesting that it's either tall or short. There's no in-betweens, there's no range, right? No range. These are all or none characteristics. Then where the flowers are, if the flowers are at the tip, or if they're at the apical meristem, apical is the tip, or in the medium, in the middle, where the flowers come out. They don't come out in between anywhere, either at the base or the tip of the branch or the twig. And then the color, finally, either purple or white. There's no range, there's no variation, like the tulips, for example, okay? So these are called oral non-characteristics. They exist in all animals and plants, pretty much, but they're not that many. They're few, all right? They're very few. For example, for the human, Mendelian traits are things like, I think this has to do with the hair in between the digits on the hand. If people can roll the tongue or not, you can roll your tongue or not, that's an all or non characteristic. And then the widow's peak, this one, the widow's peak, it's all or none. I don't know, it's associated to widows. The lobe, the earlobe, free or attached. And finally, bending the thumb backwards like that. You see some people can do that. Remember when we were kids, the kids go, oh, how ugly. They can bend the thumb backwards like that. So some people can do that, right? Okay. So these are all or none characteristics. There are very few in the human. Also, the Rh factor, this is at the molecular level. This is a good example of molecular. We either are Rh positive. Doesn't mean that we have the monkey blood. It means that we have the same protein that is detected in the rhesus monkey red blood cell, all right? Or we don't have that protein that makes us Rh negative. So that's an example of a Mendelian trait at the molecular level. Most traits are non-Mendelian. Most traits are known as quantitative traits. In other words, they're going to have a range of expression. And typically, there will be a wide range of expression. For example, what I have here is cat faces. It's all Felix, right? So Felix domesticus. So it's all the house cat, or Felix, yeah, Felix domesticus, or Felix vulgaris, I forget. The point is that these are all cats, all right? Different breeds. You can see the very wide range in faces. That's a polymorphism or a quantitative trait. Skin color, a wide range in the human. For plants, these are actually, these are tree ferns, which are considered living fossils. They go way back. They occurred in the tropics, mostly in Southeast Asia. You can see a wide range of height. Okay, wide range of height. For guppies, this is an example of artificial selection. Selecting on the tail, the pattern, the fancy guppies. Okay, wide range. These are all guppies. In principle, any one of these can fertilize a female guppy. And for those of you who are mushroom hunters and you love mushrooms, this is Boletus, edible mushroom, generally, unless it has arsenic. And you can tell if you scratch it, it turns kind of bluish. Stay away from it. At least it's going to be bitter. It could be poisonous, but if not, it's edible and it's very delicious, the Boletus, right? Okay, so this is a wide variety of Boletus mushrooms, typically depending on where they're growing. What changes their color, what changes their edibility from edible to poisonous to lethal. There's an anecdote about Darwin that with the beagle, with the ship, when they got to the coast of Brazil, what is Brazil today, and the Yanomami Indians there, he realized that they were poisoning their darts with mushrooms that were being sold in the marketplace in Great Britain, in London. Okay, so obviously the same species was poisonous in one area one geographic area, the other area was edible, which points to nurture big time on that basic plastic nature of the mushrooms. All right, so finally, we can now look at the 5 theories of Darwin for evolution to occur. These 5 theories or discoveries, okay, that species are non-constant. This is pointing out to variation, but now the variation is from generation to generation. We may be slightly taller or shorter than our parents at when we become their particular age, right? Let's say when they had us, when we reach that age, we may or may not be their same height. So there's going to be variation in every progeny, right? Non-constant. So the standard, the norm is non-constancy to greater or lesser extent of the range. Some ranges are very narrow, some are very broad. The descent of all organisms from a common ancestor, we saw that with phylogenetics. That was another insight, if you will. Descent of all organisms from a common ancestor, known as branching evolution or the tree of life, or phylogenetics, where we look at the genetic input of that. Gradualness of evolution. In other words, there are no true discontinuities, but the paucity of the fossil record. So where is the missing link, basically, right? Well, the missing link may or may not be buried in some sediment that we may or may not discover sometime. Remember, soft tissue is not going to preserve well unless there was some kind of an imprint on the very particular circumstances that we're going to get soft tissue, but mostly not, right? So there is a gradualness, but there is a continuous evolution happening. It's not discontinuous. We may not have the fossil to prove it. The multiplication of species, in other words, radiation. And in fact, we have seen now, when we look at the big picture and match up the fossil record to the strata, to the geological strata, over long periods of time, we see the cycling of radiation and the mass extinction and a further radiation because of niche availability and so forth. And typically, it could be associated with some kind of a catastrophic event, a drastic change in the environment, mass extinctions, but a few representatives survive and evolve, and then there is a new radiation occurring. So that all is called adaptive radiation. The ones who get to adapt get to survive. And finally, this whole issue of natural selection, So selection is essentially at its core, excuse me. Selection at its core is a selecting out. It's a selecting out. In other words, it's a non-surviving, right? It's not a selecting in, but it's a selecting out. And because it happens in nature, then that's the natural selection right there. Mm-hmm. And this is the only force that truly acts on variation. May act, doesn't always act, but may act. These are essentially his 5 points. Now, I'm going to go through each one of them in a, just showing briefly an example of each. But before I do that, maybe, I see it's already 11 o'clock. People want to take a break. We'll take a few minutes break and we'll come back, all right? So before I show you an example of each one of these, we're only 5 slides. Take a break, we'll come back, and then we'll look at the first 3 forces of nature, of evolution, which are, just drew a blank. Mutation, migration and drift. Okay, so let's take a break here for a moment. Pause. Okay, so I'm going to start resuming recording now again. I always forget. You people need to remind me. You know? I'm an old man. You have to have pity on me. Please. Yeah. Take turns, okay? But remind me. Thank God that I was just doing a very brief summary of the previous chapter and I hadn't started. But I was talking about mutation of DNA, which is a good segue into this other chapter, with, if you will, that we're going to see today, are the causes, the forces that cause evolution, all right? The forces or the factors, whatever you want to name it. I think Meyer has about 7 of them, but they can be clustered into these 4, which in brief are mutation, migration, drift, and selection. So we'll look at the first 3 of these today, and we'll leave selection for its own topic, because you got natural selection, you got sexual selection, and you got artificial selection. And they all play a role. So let's go right into it. Again, the diagram that we saw earlier on mutation on the DNA, right? Sticking with this diagram, we know that this is a genetic sequence, this is a genotype. So let's look at mutations. Can be a variety of different types of mutations, or it can be a process called recombination, which is going to be mostly at the level of the chromosome, or transposons, which again is going to be at the level of chromosome or chromatin. It will be in the higher order structure of DNA, the higher order structure. So let's look at, and by the way, you understand that mutations, these events are random events. The mutation itself is considered a random event statistically. And it's at the molecular level. So here we are at the molecular level. When we look at sexual reproduction or for mutations, if it's going to happen before sexual reproduction, it will be at the level of the gametes. But if it's in asexual reproduction, non-sexual reproduction, then there are a couple of ways in what is called lateral gene transfer or clones for asexual reproduction. So let's look at some examples of this. For sexual reproduction, it will affect the gametes. In other words, excuse me, the sex cells. The gametes is just a scientific word for the sex cells, egg and sperm, or ovum and sperm. The eggs produced in the ovaries, the sperm produced in the testicles. So what happens is that these reproductive organs are the ones that are affected by mutation, by radiation or other chemicals, as we'll see, or other factors. And that's why when X-rays are taken, one gets that body shield of lead so that the sexual organs are protected, and the core of the body to try to prevent mutations. Okay, so you're familiar with that. At the level of the organ itself, these little tubes that are called seminiferous tubes, they are the ones that produce the sperm. So if these get affected by mutagenic agents, such as ultraviolet radiation or some chemicals or heat, then that can cause mutation on the sperm. In contrast, in the egg, again, the ovary, if the ovary is exposed by those factors, that can cause mutation on the egg itself as the egg is developing. We'll look at reproduction in more detail in the spring, in the course on the beginning of human life, but for now, the production of gametes is called gametogenesis. Gametogenesis, all right? It's the production of gametes, and the gametes are the sex cells, once again, either egg and sperm. So those are then functionally delicate cells. They're delicate cells because if they get mutated, there's a likelihood that that will be passed on to the next generation, and that mutation will stay in the gene pool of whatever species passed on. In contrast, in asexual reproduction, non-sexual, it means that there are no gametes, there's no egg and sperm. There are different types of asexual reproduction. One type is called vegetative propagation, little shoots that come out from a tuft of grass, and that shoot buries into the soil and will produce another shoot, another tuft of grass. Okay, this is called vegetative propagation. It also happens, for example, in orchids and bromeliads when they put little babies on the side. All these are different types of clones. It also happens with animals. In lower animals, they're known as coelenterate. These are amoebas, hydras or anemones, corals, sponges also, sponge like this, a little budding off on the side, which eventually clips off and becomes a new individual. They have a lot of regenerative tissue. Binary fission is splitting in the middle, like many protozoans divide this way. It's called binary fusion. Also, a splitting similar to this could be mitosis, in that there's a splitting of cells into two. Also, fragmentation and regeneration is another example of asexual reproduction. For example, starfish, not only will, if a leg breaks off, the rest of the starfish will grow a new leg, but also if the leg can grow new legs as long as it has part of the central disc, what is called the central disc, which is this middle region here, because obviously the regenerative cells are more abundant there in that central disc. So if this arm has a piece of the central disc, then this arm can regenerate into a new individual. All right, so lateral gene transfer happens mostly in bacteria. This is a plasmid, it's a circular DNA, it's a circular DNA, little package of DNA that can be passed on by what is known as lateral gene transfer. There's a bridge that is gapped here. That's a gap that is bridged. And the DNA replicates first and then passes a replicate to the other bacterium and reassembles into the circular plasmid. So that's called lateral gene transfer. That happens, and there can be mutations along the way. There can be mutations in asexual reproduction with the progeny. Let's look at the molecular level very briefly. I don't expect you to know that much level of detail here, but different types of mutations. It could be a point mutation. A point mutation is a single base pair substitution. Okay, a single base pair substitution can cause, in extreme cases, some disease. For example, thalassemia, which is more prevalent in Europe, is changing a cytosine in the transcript. Okay. This U represents what? U represents uracil, right? And U in the RNA is a substitute for what? What letter is missing here in this transcript? C-U-G-A-C-A-G is missing T, thymine, all right? So in RNA, thymine is substituted with uracil normally. But here, C should complement with what? C should complement with G. This is actually this is actually the coding region here. because I see the transcript is the same, right? So this is already the coding region. But this cytosine was replaced by substituted by a U instead of what it's supposed to be, which is another C, right? It's a third C. What happens? That CAG codes for an amino acid called glutamine, all right, which is normal part of the protein of the hemoglobin. But U now makes this triplet be UAG, and UAG is known as a stop codon. What the stop codon means is that it stops the production of the protein prematurely. So it stops here at codon 39, and the protein is truncated. And instead of reaching its full length, right, to 146, it's truncated. See? And so it's missing a big piece of the protein, which includes the active site. And therefore, this truncated protein is a defective amino acid on the globins that form the hemoglobin, and it causes thalassemia. Some of the phenotype of it is that it doesn't bind to oxygen as well, and so, or the iron. So these people typically are anemic, and they have, they are weak, right? And they're short of breath and so forth, so it's a condition. All because of a single point mutation on one nucleotide of the DNA, of the transcript, on region 39. It has been mapped out to that particular region. It's amazing. Rarely does this happen, but it does happen. I'm not sure that cystic fibrosis is also a point mutation. I'm not sure, but I think cystic fibrosis is also a point mutation. Okay, we can have deletions also. When you have a deletion, it's a problem because then it changes the reading frame. It shifts the reading frame. So for example, here, it just gives a very simple reading frame. It turns out that that little example that I was using, CAT, codes for histone, which is one amino acid here, HIS. So that triplet CAT on the DNA codes for one amino acid known as histone. And so here they just made a simple sequence of the same. So you see it's a little polymer of histone amino acids, right? So CAT, CAT, CAT. Now, if 1A is taken out, it turns out that the reading frame changes, and this first one, CTC, will code for leucine, which is a different amino acid. But then the next one will go back into a pattern of ATC, and ATC codes for isoleucine, ILE. ATC, ATC, ATC. So you see, it changes the reading frame. And so it changes the sequence of amino acids, which will make a different protein, or no protein will make a nonsense, okay? Many times a point deletion will make a nonsense code that it doesn't really code for anything. So again, it will cause a premature truncation or a miss, a protein that is not folding correctly, doesn't have the proper amino acids, the proper building blocks. So this to point out that there are different types of mutations at the level of each individual nucleotide, level of each base. Continuing then, other types of mutations, sometimes there are segment deletions, a whole segment can be deleted out, or inversions, duplications. One segment here is the two alleles of a chromosome. These are two alleles of a chromosome, right? This is when the chromosomes have replicated, preparing for the cell reproduction, preparing for mitosis or meiosis. And there's a break and reattachment. So this is a duplication on one of the alleles. And the other allele will have a truncated end. Sometimes the genes are inverted, the sequence is inverted. So these are different types of mutations that can occur. They all occur at the level of the DNA. And they can be caused by any number of factors. I mentioned UV, okay? So UV is the main source of mutation, which is blocked out by the ozone layer, which is O3. O3 is only stable at high altitudes, and that's why that ozone layer is very important. If we have a hole through the ozone layer, it means that more ultraviolet radiation is going to come through. But also chemicals can cause mutations. These are called mutagens or mutagenic chemicals. There's one that's called acridin, for example, that fits inside the DNA molecule and generates some kinks there so that it will alter the sequence of the DNA. And again, I mentioned heat. Heat can break these bonds of the DNA. And you know, one of the factors for, it's one of the possible reasons of why the testicles in mammals, in male mammals, are hanging outside the body, because the body temperature is actually too warm and it kills the sperm. And if it doesn't kill the sperm, it may actually cause mutation on the sperm, which will make that sperm non-functional or non-capacitated to fertilize. So heat is definitely a mutagenic factor. Okay, another aspect of mutation that is at the level of the larger of the chromosome when the cell is replicating is this event called crossing over. Crossing over, or another source of variation that is put. That's why Meyer makes a distinction. I see now why he makes the distinction between mutation and recombination, because this is an active process that occurs in nature internally by the chromosomes splitting and sharing bits of chromosomes, whereas mutation is driven typically from some external agent that is going to cause a chance mutation. So I see why now he puts recombination as a separate category, but I'm lumping them together. Definitely at the molecular level, this event of crossing over occurs in the process of meiosis and the process of gametogenesis before the second split of the cells here. There's a first split and the chromosomes are still in pairs. In this pairing, this is where the crossing over can occur, so that when the second split happens, those chromosomes have already recombined, and one allele will carry a segment of a chromosome from another chromosome, from its sister chromosome, the chromosome pair, and vice versa. So they exchange pieces. You see, of the 4 resulting chromosomes, these 2 are recombinant chromosomes, and these 2 are non-recombinant. They are originally intact, but the ones that undergo the recombination process, they're called recombinant chromosomes. All right, let's see. So we move forward. Yeah, so this occurs in the process of gametogenesis, the production of gametes, and it is known as an independent assortment of alleles. Independent assortment, in other words, here are two possibilities. These are the chromosome pairs, right? This is, let's say this is pair 1 and this is pair 2. This particular species only has 2 pairs of chromosomes, or a total of 4 chromosomes. This is pair 1 and this is pair 2. There are 2 possibilities when these cells split. There are 2 scenarios, if you will. This is possibility A, this is possibility B, all right? This is called the assortment, or the distribute. It's like distributing a set of cards, but you only have 2 cards. You can distribute the ace first and then the king, or you can distribute the king first and then the ace, right? So you have 2 possibilities. So this is one possibility here, where the, let's say the blue chromosome 1 and chromosome 2 stay together, and the red chromosome 1 and chromosome 2 stay together. That's one pattern of distribution. The other pattern is that they're flipped. You get 1 blue chromosome and 1 red chromosome, and then on the other cell you have to have, by default, you have to have 1 red chromosome and 1 blue chromosome. So you see, you have either one of these possibilities. There's a 50-50 chance that this would happen. Oh, sorry. Then on the second split, it's locked in already. So meiosis is a double split of cells. On the second split, they're locked in, because if this cell has only blue chromosomes, then the daughter cells can only be blue chromosomes. And the same is said for the red. If it only has red, well, if this one has only blue, then this other cell can only have red. But here, it's different, because there's 1 red, 1 blue. The split is going to maintain that recombinant state, if you will, red-blue, and also for the other one, but on the complementary pair. The bottom line is that when we look at the distribution, see this, because this is gametogenesis, right? This is a double splitting of cells. So this is meiosis. Meiosis is the formation of gametes. At the end of the day, these cells down here, these are gametes. So you think of these as either egg or sperm. You see how the pattern changes. For this set of sperm, chromosome 1 and chromosome 2 are the same allele. Okay. But on this other set of sperm, chromosome 1 and chromosome 2 are from different alleles, are from the sister alleles. And there are, that's why they call alleles, because they may have localized mutations between the red and the blue. In other words, the red and the blue of the same chromosome pair. Let's say that this is chromosome pair number 1. The red and the blue have slightly different sequence at the local level, all right? They have slightly different sequence at the local level. You can think of it as a genetic fingerprinting, if you will, that distinguishes the red from the blue chromosome of chromosome 1 of the pair. So it will make a difference. At the end of the day, what I'm trying to say is that it will make a difference which one of these sperms fertilized, where it was from this set A or from set B. Because set A will carry these particular characteristics, and set B will carry this particular specific characteristics at the level of chromatid, at the level of the allele. Okay. Again, to point out that perhaps it's a little bit too much detail at this point, but the issue here is that crossing over, that recombination, can also add variety, is another force of evolution that evolution will use, if you will, to effect a variation on the progeny. The third event that happens at the chromosome level is something called transposons. Transposons are segments of DNA, movable segments of DNA that jump around within the chromosomes. They jump around within the genome, all right? And it's pretty significant. It's estimated to be about 44% of our DNA, of our genome, is transposable elements, transposons, these segments of DNA that have jumped from one region to another. They are considered to be the remnants of viruses or fragments of virus. Andrina?
Is that the same thing that they call jumping genes, or that's not? I know about what a jumping gene is.
Yeah, I don't know right off. However, I'm thinking that transposables are not necessarily genes, they're just sequences of DNA, so they may or may not contain genes. You see? A jumping genes is specific for coding region. So this also includes non-coding region. So again, thinking out loud, I would say strictly speaking, it is not, because jumping genes is specifying coding region. But transposons can also be non-coding. In fact, they're estimated to be perhaps remnants of viruses, which are coding for the virus, but not for us. But they were integrated into the human genome, into the human DNA. I'm going to show you a little film in a minute on how the process happens, a 3-minute film. Oh yeah, so there it is. From viruses. They can be beneficial, deleterious, or neutral. Most of them are neutral, depending on where they fall, if they disrupt the sequence, if they disrupt the coding region. But considering that 97% of the genome is non-coding, the chances of them landing in a coding region is slim, is very slim. So thank God, these guys can move around in the genome and they don't really have an effect. Typically, they're neutral, right? Or if they jump into a coding region, most likely they're going to be deleterious. Very, very seldom they may be beneficial. And then, of course, they're going to enhance survival. If they're beneficial, somehow they're enhancing some kind of a biochemical pathway. Okay? That's what it means to be beneficial. What do we mean by beneficial? That is enhancing some kind of a pathway. I think I have a little bit of examples further down. But let's look for a moment at this video. How it explains the transposons. Of course, there'll be a little ad first. 1 minute, it's over. Oh, that's another one. Okay, now skip it. And volume again. Let's stop this. Get the volume up here. All right. Did you notice any difference between the volume on the videos or my regular volume? I don't know if it made a difference on the...
Transposable elements, which are found in virtually all cells, are segments of DNA that can move from one location in the genome to a target sequence in another. A simple type of transposable element called an insertion sequence, or IS element, is only 700 to 1500 base pairs long and consists of 2 basic parts, a coding region for an enzyme called transposase and 2 short inverted repeat flanking sequences. The cellular machinery transcribes the transposase coding region and translates it into the transposase enzyme. Transposase catalyzes the transposition process. The transposase randomly selects one of many possible target sequences where it will move the insertion element. The IS element, depending on the particular type, may enter the target sequence by one of 2 general mechanisms: replicative transposition or non-replicative transposition. In replicative transposition, transposase nicks the transposable element and the nicked ends attack the target sequence, making a staggered cut in the target DNA. DNA polymerase fills in the large gaps, and DNA ligase seals the remaining nicks. A recombination event resolves the 2 molecules. As a result of replicative transposition, the cell has a copy of the transposable element flanked by duplicated target sequences. Non-replicative transposition is sometimes called cut-and-paste replication. In this process, transposase brings inverted repeats and flanking DNA together. One phosphodiester bond is cleaved on each strand at opposite ends of the IS element, yielding 3 3' OH groups. Each 3' OH end attacks the other strand of the double helix, creating a new bond that results in hairpin structures. Host carrier DNA is ejected, and the hairpins on the IS element are reknit and attack target DNA. From the 3' hydroxyl groups, the cell's DNA polymerase molecules fill in the gaps, and DNA ligase seals the remaining nicks. In this way, the element jumps from one DNA molecule to another. To summarize, in non-replicative transposition, the transposable element is cut and pasted from one DNA molecule to another. In replicative transposition, the transposable element is copied to yield 2 transposable elements, one in the original position and one in the target DNA.
Okay. So one thing I want to point out about this is that it is extremely mechanical, as you can see. All right, it's very mechanical, and it's down to the molecular level. But these processes are happening in the DNA. They're happening all the time, to the point that about 40%, over 40% of our genome is composed of these transposons, which in principle are foreign DNA that has been integrated, incorporated into our own DNA, into our own genome. And the prevailing theory is, again, from these, from viruses, which is essentially a DNA or an RNA filament with a capsule, with a protein capsule. So just to show briefly the cycle of viruses, these are photographs. These are electron micrographs taken with electron microscopes. Imagine this is 100 nanometers here, so it's almost getting down to the molecular level. This is very high magnification of different types of viruses. But the general life cycle of the virus is first the lytic phase, or the infective phase, and then there's the DNA or the RNA of the virus. By the way, phage or phage is another name for virus or phage or phage, depending if you're American or British, right? It goes in, first it's a plasmid of its own, circular DNA, but then it gets integrated into the DNA of, in this case, a bacterium, and it becomes what is known as a prophage. A prophage is integrated into the bacterial DNA or the cellular DNA. And that cell can undergo many divisions and maintain incorporated this plasmid, or at some point after many divisions, the plasmid may jump out again and start coding for the proteins that are needed to make up the virus. And then, so when the DNA incorporates into the bacterial or the host DNA, that's called the lysogenic cycle, lysogenic. And then at some point, that DNA will pop out again, make code for its own protein, which it sequesters the machinery of the host cell, the protein machinery. And that's why viruses are considered, they're not considered to be really living, because they don't have the capacity to reproduce on their own. They need the protein machinery, the replication machinery, and transcription and everything of the host cell. Okay. And then when they become very large in number, eventually they'll burst the cell open. And another name for bursting a cell is called lysis, lysing the cell. And that's why this is called the lytic phase, because the cells burst, and from one virus, many hundred thousands, millions of viruses come out, and each one of those potentially can contaminate another cell in the body. So now we're dealing with a viral infection here, rising very fast exponentially. But this is just to show that the standard mechanism for viruses to integrate into the host genome and multiply, and at some point pop out and reconstruct the virus with the proteins, because it's a protein code. And when they get to be too many, they pop out of the cell, break the cell, lyse the cell, and that's what causes the infection, the reaction and the flaring up. And that's why viral infections tend to have a flaring, and for the classical one is malaria, right? That it flares out and then it slows down because the virus is cycling from lysogenic to lytic cycle. Anyway, just to show that viruses today have this innate mechanism of incorporating their DNA or DNA into the host DNA, and it's called a prophage. Okay, so this gives you a little bit of a summary of the first force, or one of the first force that occurs by randomness. We saw the randomness in the ultraviolet causing mutations or recombination event in scenario A or scenario B. And then also transposons. It's basically junk DNA that has been accumulating, fragments that have been accumulating into a genome over the eons, over millennia of time. All different types of forces that occur at the molecular level. Let's back out a little bit to the population level for a moment, to the individual level, and let's look at gene flow. Gene flow is also known as migration, and there are two movements in migration, right? Either immigration or emigration, correct? Moving into an area, into a geographical region, or moving out of the geographical region. So immigration or emigration. All right, so let's look at that. I have some examples here. It's at the level of the individuals within the population, but it's always a population involved, okay? For example, let's look at the zebra. We can't really, so we see that these zebra are migrating, right? Even if it's a still photograph, we intuit that they're moving, and they're moving from right to left. We cannot tell if they're immigrating or emigrating, but they're moving through the savanna of Africa, most likely. And maybe they're fleeing a lion or a pod of lions or whatever, but they're definitely migrating, either immigrating or emigrating. If they're fleeing from hunters, then they're emigrating. If they're going for pastures, they're immigrating, right? Depending on what's the function, what's the behavior. But these zebras look very much alike, right? And we can say, well, yeah, they're all alike. They're all the same zebra, but the pattern varies. Look at the pattern. So this is a close-up of this region here. Should have put a little boxes here, but this, you see these 4 zebras here? See these 2 heads? These 2 heads are these 2 heads here, the muzzle. Okay, when you look at the zebra population as a whole, you kind of miss out, we miss out on the pattern, on the individual pattern of the individual. But the variation is definitely at the level of the individual, because you can see this blow up here. Let me enlarge this a little bit. How do I get over? Here it is, the bar. And the other bar. We're going into the next slide. Up to the next slide. There. Okay, so let's look at this pattern a little bit. Look at this zebra on top. See the pattern here of stripes? 1, 2, 3, 4 stripes before the V. This one, 1, 2, 3, 4, 5. Okay, there are 5 stripes here. There are 4 here. And the pattern is different here. You see how there's only 1 V here, but there are 2 V's here. There's 1 V and there's another V. So see how the pattern is definitely different at the level of detail between these 2 zebras. Look at the neck, right? So you can see the pattern about these 2 here. Look at the pattern here and the pattern here. So there's a general pattern that they'll share of stripes, but there's a specific pattern that varies definitely from zebra to zebra. Compare this rump to this rump, for example. And so we don't know what the animals are selecting on. It could be that this is a selective advantage or disadvantage on the pattern for sexual selection. Probably not for natural selection. Most likely the banding pattern is estimated to be in nature in general to be a confusing pattern, so that the lion or the tiger, whatever is going, the cheetah is going after these, is when you see all these moving and moving like that in a sequence, it's very hard to focus on an individual zebra, right? Because of course the lion has to hunt down an individual zebra, cannot hunt the whole group or half a zebra or 3/4 of a zebra, has to hunt an individual, has to focus on one individual to hunt it down. So that's one example that has selective advantage. But also back to migration. So this is about migration, right? Meaning that it depends on which zebra got into what area and mated with what other zebra. That particular pattern is likely to be passed. So I don't know if typically for mammals, the typical pattern is the harem, where there will be an alpha male and several females. So that way he gets to fertilize several at the same time, and that it increases the chances of the survival for just by volume, by number of offspring, because the one male will have actually several females in gestation simultaneously. So that's a standard pattern pretty much for mammals in the natural world, except for humans, right? And there are a few examples also in the natural world of monogamy, but they tend to be the exception. It happens in fish, it happens in birds, and it's supposed to happen in humans. All right, so if let's say that this is a cluster of females that belong to this alpha male here, I'm just saying this one darker, maybe the alpha male. So what is he selecting on? He may be with the migration, it may be that the females in the back did not get to migrate to the same place where this male migrated. So obviously the mating is going to be at a very localized level, and it's just the individuals that happen to be from the large population, the individuals that happen to be in that particular localized region that get to mate. So migration will make a difference. It's one of the forces that will act, because that male will mate with these females and not with these other females that are in a different migration route or in a different geographical area. Okay? So migration will affect, and when we take it to the genetic level, that's called gene flow. In other words, for geneticists, we tend to see this individual species as just bags of genes, if you will. They're carrying back genes that may or may not be expressed at the phenotypic level. Okay, but looking literally under the skin down to the gametes, to the sex cells, what we're looking at is the genotype, the genes, the particular genes that they carry. Here's another example in plants, because it also happens in plants. Typically, when we talk about migration, we think of animals that are motile, right, that can move around, mobile. But no, it also happens in plants. And this is one example here. What am I trying to do? Make this a little bigger so that you can see. Where am I? Here's the bar. Okay, so What's your intuition? How many populations of grasses are in this, on this shoreline here, in this particular photograph?
At least two.
At least?
At least two.
At least two, right? At least two, right? Because we can see that because they're individual species, that this type of grass or this type of creeper is a different species from this type of grass here that grows in tufts. This one grows more of a creeper. I don't even know if this is a grass, can't really tell. But this is more of a creeper. And this is a population of that plant. This is a different species, definitely a different species, right? You see the pattern of distribution is also more scarce or sparsely populated. However, because there is a ridge here, there's a ridge between these two little dunes, okay? These may actually be two populations of the same species. So we can have two different populations of the same species. What we cannot have is one population of two different species, okay? So you can see here is a population of this species of plant, I'm going to call it plant A, and here is another population of that plant A. But this, to stay with the analogies, this could be uppercase A and this could be lowercase a. In other words, this population here may have certain characteristics that it makes it more adaptable than this one, all right? Simply because of its location. This grass here is more sparsely populated, and you can see that definitely they are influenced by the local geography, all right? Barely making it here. And what's your intuition about the root system that is underneath the sand that we cannot see, all right? But we can intuit that all these have root system. Would you expect this to be a very rather shallow and small root system, or a rather deep and extensive root system down there? Most likely a deep and extensive root system trying to absorb every little bit of humidity that they can find underneath the sand, away from the surface where there is more evaporation. Now you see that the pattern of migration is different. Oh, that's what I wanted to show specifically. This grass is migrating here, or this plant is migrating on the surface of the sand, whereas this other grass seems to be migrating underneath by they're called what, rhizoids. They're little shoots or tufts that occur underneath the sand, all right? So this species has a different pattern of migration than this other species. The one species migrates on the top of the sand by shoots, the other exposed shoots, whereas the other species migrates underneath the sand, again by these rhizoids, but a different particular mechanism. So migration also occurs in plants, and it can determine how, when they become geographically isolated by a little ridge like this, even this could be sufficient, because you can see this environment is so harsh. The external factors are so acting here on these plants, that over a period of time, this population of grass may develop characteristics that are different from this other population. And even though they belong to the same species, at some point, if you put them together, they will not interbreed. They will not interbreed. They will not recognize each other, because it will go down to the molecular level. That's the beginning of speciation. If that selective pressure continues over a long period of time, they can diverge enough to become first different varieties and eventually perhaps different species. So long periods of time are definitely involved in this. Okay, another type of migration that, just by the size, by the range. It could be very localized, or it could be very vast. I'm giving you two extreme examples here. Anybody know what this is? It's a sloth, right? It's a sloth, and they occur in tropical regions. I've seen them crawling around in Panama, for example, the jungles of Panama, and Central America. And there are two-toed sloth and three-toed sloths. This is a three-toed sloth. You can tell by the nails. It's got 3 nails. Then there's another one that's got only 2 nails. So there are two-toed sloths and three-toed sloths. It is estimated, it's been documented that these guys, and this is the mom, and this is the offspring here hanging on, seems to be very happy by just hanging on and letting mom do all the work. But basically, these are mammals, kind of primitive and slow, and it's estimated that they only move a few meters from the place they were born in their whole lifetime. Okay, very slow. That's where the word sloth comes from. They're very slow growing, very slow moving, very slow growing, moving. Typically hang there, they're herbivores, and they just hang there and eat the leaves. In Central America, sometimes they get to cross the road there. They get on the road in the urban area, in the rural areas, and they'll take hours to cross that road. And you see the traffic backed up, all right, or sometimes they get run over, but they're so prominent. And then the next sequence is that the kids, the poor kids, find them somewhere crossing the road, trying to cross the road, and they take him and they grab him, and then the kid stands on the side of the road selling the thing. And they'll stand there and they have the sloth around them, okay? And people will buy them. They're not supposed to, they're protected and so forth, and the kids are not supposed to pick them up, but they do. Anyway, just to show that here is a mammal with 4 legs that moves very slowly from the native place, so that migration will be minimal from population to population. But still, they would make distinct populations, just maybe one tree is one population, and adjoining trees will be other populations of the same species of sloth. In contrast to that, on the other extreme, vast territories from something that is grounded with roots, right? So the level of individual, this population of conifers here, or pine trees, each one of these individual conifers is grounded to the ground and is moving even less. The individual tree is obviously moving less than the sloth, right? The sloth has more motility. The individual sloth has more motility than the tree. But the population as a whole, because these are plants, they produce seeds, and these seeds happen to be airborne. Okay, they're propagated by wind, and they're fairly light in weight on purpose, so that they can expand a vast territory of many square kilometers, hundreds of square kilometers or square miles, in propagation of these individual conifers, shooting out their seeds, or just letting the seeds fly out with the wind patterns through the mountains, and they will have a much wider dispersal rate and range than a mammal, which in principle is capable of, moving more than just a tree that is fixed to the ground. Okay, so you can see that migration also, there's a wide range, it's a wide variety of styles and patterns of migration. Many are seasonal, but some are not seasonal. Another aspect about migration is that there's a dynamic state occurring, a dynamic state which will make a difference between the core and the periphery of the population. So where the individual is within the population will make a difference. So if the individual is toward the core of the population, it will tend to be preserved, but there's more exposure on the edge of the population to other factors. And so here is a population of, what are these, daisies, maybe, right? intermingle with some other flowers. But so we can see that the core is more solid, monoculture of daisies, whereas the edge here has a greater variety of other species. So there's an intermingling of species. So what I'm trying to say here is that the species at the edge have to deal with a different type of competition than the species at the core. The species at the core is an intra-competition, but here is an external competition, mostly for grounding, okay? So in principle, even the less vital genes may get to survive at the edge because it's a different type of competition than the core, where those genes are competing with more of the same gene or more of the similar genes. It's the same happens at the microscopic level. For example, with the pattern of growth of bacteria, here's a Petri dish that is super contaminated with bacteria growth. Here is an enlargement of any one of these spots. The core of the bacteria tend to be more protected than the edge. And you can see how the edge is graying. Why? Because this plate has been treated with penicillin or some antibacterial that is killing, and it's going to kill the bacteria on the edge first. Eventually, the effect of the antibiotic will wear out as it's being used up and penetrating, and maybe the core will survive, and only the edge got killed. So you can take migration down to the microscopic level, how it may have an effect on a population, even for survival or not. So again, the periphery tends to have more variation than the core in any given population, whether it's at the microscopic level or the microscopic level, whether it's at the animal or the plant level. Also, with regards to plants, the edge, typically they're known as pioneer species. They're conquering an area that is not yet with habitation with life. And these are mostly lichens and mosses because they produce acids that will start eroding rock, will start dissolving rock, which tends to be on the basic side of the alkaline side of the pH scale, will be eroded, corroded by the acid that is produced by lichens and mosses, lichen, which is a combination of algae and fungi. And those are the pioneer species. Then as more soil gets to accumulate in the area, partly from detritus, the dying of these species and becoming part of the humus of the soil, bigger shrubs and plants begin to establish in the area. And you get what is known as a series of successions, which tend to move toward a more established ecosystem. And there will be larger trees, fewer species of larger trees, both conifers and deciduous trees, until eventually, if the climatological conditions remain the same, more or less the climate remains approximately the same, you end up at the very end with a climax community which has 1 or 2 or 3 dominant species. And there will be the largest trees and the oldest trees in the forest. And then there is no more succession. There's no more succession because colonization has stopped. These trees, that's why they're called dominant species. Year after year, decade after year, millennia after millennia, they will continue to be the same species of trees, okay? Unless there is some kind of climatological change, and then it may or may not have an impact on that forest. And I point this out because our slash pine trees that we have on campus, they are the dominant species of this climax community. And we're trying to protect them, but it's, they are the only pine trees, they are the only conifers that exist in South Florida, in the peninsula, whereas the peninsula used to be covered with these pine trees back before modern civilization, modern metal tools, about 120 years ago. And now it's less than 2% of the county. Okay, so they're disappearing slowly but surely, and they're not coming back because most of urbanization. So we're trying to preserve our forest here, and it's a challenge. Okay, so let's look at the third force, which is drift. So we got gene flow. Gene flow translates into migration, right? Gene flow is at the level, at the genetic level. Migration is at the level of population. So I'm going kind of back and forth here between the individual and the population. The third force now that might cause evolution, the third force is called genetic drift. And this is a random event. A random event in science is called a stochastic process. All right, it's just a technical term. You run into it in the literature. A stochastic process is a random event. It's a chance event, like flipping a coin. What are the chances of getting heads or tail? On average, 50-50, right? You may get 3 heads in a row, but then somewhere down the line, if you continue to flip that coin, you're going to get 3 heads somewhere else at some other flips. So that when you start averaging up all the trials, you end up with half the time, on average, half the time was heads and the other half was tails. So that's just a random process because there's 2 possible outcomes. So it depends on the possible outcomes. In there are 3 possible outcomes, then on average it's going to be 1/3, 1/3, 1/3, and so on and so forth. You can continue to play with the math there. The more outcomes you have, the more variety of outcomes, okay? But they will be, normally, on average, it will be evenly distributed. That makes it a chance event. Okay, so we can look at the population of fish here, and there are at least 2 very visible population of fish. One is the blue tang, and the other one is the yellow wrasse. You can see the blue tang are mostly blue. The yellow wrasse are mostly yellow. You can see that just by the eye, there tends to be more variety of color, of coloration in the yellow wrasse, to the point that some are not yellow at all, they're orange. Whereas the blue tang tend to be blue, some darker, some lighter blue, but that could also have to do with the lighting. But there tends to be less variation of color in the tang than in the wrasse. That's just an observation as we look at these 2 populations. Okay? So genetic drift has to do with the sampling error. In other words, you can see that this photo is taken here, right? So this photo includes a sample of the blue wrasse and a sample of the the blue tang and the yellow wrasse. This photograph, this frame, all right, when the diver took this photograph, this is a sample of the blue tang and the yellow wrasse. But this is a coral reef, right? You see the coral reef down here? So we can assume that this coral reef is broader, is bigger than just what's fitting in the picture, in the photo. So this school, they're called schools of wrasse and tangs, is bigger. The population of this blue tang is bigger. For example, here we just see half a tang, right? The half of it. So the other half of this tang is not in the sample. Here's another tail of another tang over here that's facing that way. So there are more tangs in this population than what we're seeing. So this is just a sample. The same can be said for the wrasse. There are more wrasses in this coral head than the ones that are seen in this photograph. But the sample only involves the ones that we're seeing here in this photograph. There's a migration, there is migration in and out of this, so it's a dynamic state of migration in and out. But the actual drift is which one of these tangs get to mate, for example, or recognize each other, and which ones get to be taken out with a net for an aquarium, get to be fished out, if you will. Let's say that this diver is also a collector because he has a, or she has a saltwater tank at home, aquarium, and they're going to try to grab some of these fish. They'll be grabbing some and not others, so that's a random chance. What's the intuition? The intuition is that on average, the slower fish is the one that got caught in the net. On average, the slower fish. So being faster swimmer can be a selective advantage, right? That's just a natural intuition. But it also happens that the net is over here and the net is not over here. So I'm catching this tang, no matter how fast or slow this tang is, if my net is over here and not over here, well, bad luck. This tang happens to be in the wrong place at the wrong time, or the right place at the wrong time, where you put it, because the net is here and the net is not over here. That's just a chance, you know, because just by looking at this fish, I cannot tell from Adam which fish swims faster than the other. So I'm just going to make it a grab, but my net has to be in a particular local place and not in another place. Okay, so that's the drift. The genetic drift is just simply that this pool of genes happens to be where the net is. So this pool of gene happens to be selected out and will no longer compete in the general pool of genes because this fish got selected out just by chance. And that's the drift, okay? That's the drift. It just points out that every sample has some error in trying to represent the whole population. Every sample is limited. And another example of this is called bottleneck or genetic, the drift. And you can see here in the marbles, let's say that this is an even distribution of marbles, all right? Even distribution meaning that there is 300 yellow marbles and 300 blue marbles in here. But when they go through a bottleneck effect, which could be geographical or climatological, et cetera, by chance, it happens to be that, is there an even distribution of marbles here?
No.
No, we only got two yellow marbles and 2, 4, 6, 7 blue ones. It happened to be by chance because those are the ones that went through the bottleneck. Okay, so locally here, this little local sample was not as fully representative as the whole population, as a whole jar. So these are the ones that made it through the bottleneck. The bottleneck is a selective activity, selected environment. Again, it could be a geographical issue. It could be climatological, etc. So from this surviving individuals, a new population will develop. What's your intuition? Are there going to be an even number of blue and green, blue and yellow? No, because you start out with, yeah, there's going to be more blue. On average, there's going to be more blue when these individuals start reproducing and so forth. You end up over next generations more blue than yellow because there was a bottleneck effect. And the bottleneck was not selecting, they're both the same size. So there was not selecting on size, it was not selecting on color because the bottleneck is blind, if you will. It's just a chance of the local population, the local sample, when the bottleneck effect happened. One more example, one last example here is you got the blue beetles and the brown beetles, and the green beetles are all the same size, but the brown beetles have 2 different sizes, right? And this careless fellow came around and stepped on, it happened to step on green beetles, but not the brown beetles. So reduce the population of green beetles and not the brown beetles. I think this example would have been more interesting if he had stepped on the little beetles or maybe the large brown, because that would have eliminated large from this population. Whereas here, these 3 are all large. What one possible effect? No, okay, I get it. What this is trying to illustrate is that when you just have one individual green beetle, who is that green beetle going to reproduce with? Nobody. So this guy is going on extinction. In other words, from stepping on a sample, from cutting out a sample of the whole population, it may leave the population so impaired that that population will become extinct. And so there's going to be a displacement of the green beetle by the brown beetle by some stochastic event, by some chance event. Okay, I mentioned that these could be biotic factors or abiotic factors. In other words, it can be competition for some terrain, or it can be a phenomenon that is non-biological, non-biological. For example, here there is grass. We can assume that there is the same species of grass inside the cow pasture and outside the cow pasture, which is just held by this fence, right? Probably the fence has a little bit of electricity to put a little shock and prevent them. So they develop a behavior to stay away from the fence, all right? But you see, as far as the neck can reach, then that's going to be part of the cow pasture also, even though it's technically outside of the fence region. So the grasses that were on the fence, on the edge, are going to get eaten up. But if they're further away, than the reach of the neck, they get to survive. So the selecting factor here is a biotic factor, meaning that is the cow that is putting on the selective pressure on the edge of this grass. Again, the intuition is that the grasses are the same species inside and outside of the pasture. So in this case, what is putting under pressure, the selective pressure, is a biotic factor, it's an animal, it's a living thing, right? And this poor tree that got hit by lightning, which I can tell you, guarantee is not going to survive, because we had one happen here. This tree is an abiotic factor, non-biotic factor. It happens to be a climatological factor, at lightning, okay? But the selective pressure on drift can happen either by biotic factors or abiotic factors. And this tree just happened to be the largest one, which was typically a climax community, and for getting to be the largest one, got hit by lightning. This random process also occurs at the molecular level, at the genetic level, with dominant and recessive alleles. So of these, I guess, ladybugs, right? They have a range of pigmentation, these ladybugs. Some get selected out, the dominant, which is AA, capital A, and capital A. Some get selected out by rockfall. Then the heterozygous, which is a blend, you see, this is a polymorphic trait, meaning that it causes a range of color. This didn't get to find a mate for whatever reason. And so it will skew the population toward the recessive, actually, which is white. In the next generation, we can see that the red is going to disappear because the red doesn't have anyone to mate with, whereas the others, it may even out somewhat because the lowercase a, which is recessive, now just by this stochastic process becomes more abundant proportionally because of the disappearance of these other ones. Okay, I think you get the point. Overall then, we have these four forces: mutation, migration, drift, and selection. But the one that really acts on the evolutionary force is going to be selection. The one that really acts on variation is going to be selection itself. So it includes, selection includes, it's a more global one, okay, which I'm leaving for the next lecture. So that's the end there. And good. Questions, comments? No, everybody gets the process. Okay, hopefully this will sound a little louder. I tried to make an effort to be closer to these microphones. I'll send you these PowerPoints As soon as we're done here, you have the diagrams. I also sent them last night electronically, so you can work with that diagram to make your summaries, all right? So everybody okay? I'm gonna pause the recording now, stop the recording. So thanks again.